All right, welcome biology students to today's explainer. We are diving straight into a literal microscopic battleground today. We're going to break down the viral infection cycle, which honestly is essentially the ultimate biological hostile takeover. You see, when a virus encounters a host, it is not just some passive event. No, it's an active, insidious invasion. And getting a solid grip on this process, it's absolutely fundamental to understanding how disease actually spreads at the cellular level.
Okay, so a quick look at the syllabus for today. First up, target acquired. Then, breaching the host membrane, followed by commandeering cellular machinery. After that, we'll look at the viral assembly line, and finally, wrap up with exit strategies and clinical impact. Let's get to it!
Now, before we really zoom into the nitty-gritty microscopic details, let's build a quick mental scaffold. The entire life cycle we're about to explore really just boils down to three main macro stages: infiltration, replication, and release. I want you to keep this simple roadmap in the back of your mind as we get into the complex mechanics, because literally everything a virus does fits perfectly into one of these three buckets.
Section 1. Target acquired. Viral attachment.
So this brings us to a super important concept: viral specificity. A virus cannot just bust into any random cell it bumps into. It's actually way more like a guided missile. The whole infiltration process kicks off with this highly specific mechanism, where the virus has to meticulously pick the exact right biological lock on the outside of the cell. They are incredibly selective. They only recognize and bind to very particular proteins or receptors on a specific host cell. Basically, if there's no match, there's no entry. Infection simply cannot happen. To anchor this in reality, let's look at a famous recent example: the coronavirus, SARS-CoV-2. It specifically targets what's known as the ACE2 receptor. And because these ACE2 receptors are super abundant in the cells lining the human lungs, the heart, and a few other organs, well, those are the exact tissues the virus goes after. The virus uses its outer protein coat, those famous spike proteins, to attach to the receptors. It's literally like a key slipping into a keyhole. By finding that perfect fit, the virus essentially tricks the host cell into dropping its guard.
Section 2. Breaching the host membrane.
Okay, so once it's attached, the virus has to actually get its dangerous payload inside, right? And it typically does this through one of two ways. First, we have membrane fusion. This is where the virus directly merges its own outer envelope right with the host cell's membrane, basically sliding through the barrier seamlessly. The other method is endocytosis. In this scenario, the host cell is tricked into swallowing the virus whole. It completely engulfs the viral particle, pulling it inside via a tiny cellular bubble.
Now, once the virus has successfully breached the perimeter, regardless of how it got in, this absolutely crucial step has to happen: uncoating. Once inside, the virus drops its disguise. It sheds that protective outer protein coat we just talked about. And why? Well, because it needs to release its internal payload. That's its genetic material, usually either DNA or RNA, and it dumps it directly into the host cell's cytoplasm. The second that genetic code is free within the cell, the hostile takeover is officially on.
Section 3. Commandeering cellular machinery.
You know, the really dark irony of this whole process is that the host cell doesn't even realize these new viral instructions are an alien threat. It just blindly reads them as if they were its own natural blueprints. So the host cell completely stops performing its normal, healthy biological functions. It is essentially held hostage. The virus forces it to use its own cellular machinery, specifically the ribosomes and enzymes, to start producing viral components instead. So the poor cell is literally doing all the heavy lifting to build its own destroyers, perfectly replicating the virus's genetic code and manufacturing all these new foreign viral proteins.
Section 4. The viral assembly line.
This transformation is rapid and it's totally chaotic. The host cell is no longer a functioning part of a human lung or heart. Nope, it's been converted into a dedicated, highly efficient virus-producing factory. And the end result of this factory is what we call a virion. A virion is a complete, newly assembled viral particle. It's fully armed and totally ready to exit the current cell to go infect others. You can basically think of virions as the finalized microscopic products just rolling continuously off this hijacked assembly line. And the sheer scale of this production, it is completely staggering. This process is insanely fast, operating with, honestly, a terrifying biological efficiency. A single virus entering a cell can force that host to churn out thousands of copies of itself in just a few short hours. Literally, thousands. The cellular machinery is just locked in absolute overdrive, entirely devoted to mass-producing the enemy army.
Section 5. Exit strategies and clinical impact.
So eventually, these new viruses have to leave, right? They use two primary methods to exit, and this determines the immediate fate of the host cell. First, there's lysis. In this method, the host cell gets so ridiculously packed with newly assembled viruses that its structural integrity just fails, and the cell actually bursts open. This kills the host cell immediately, releasing a massive flood of new virions all at once. The second method is budding. Here, the virus kind of pushes its way out, stealing a little piece of the host cell's outer membrane to use as its own new envelope. Budding lets the virus exit gradually one by one, which means it often doesn't immediately kill the hijacked cell.
Now, while budding acts as a sort of slow drain on the cell, the clinical impact of lysis is often incredibly severe. When host cells burst and die en masse during lysis, it's not just some microscopic event happening in a vacuum. It translates directly to macroscopic clinical realities for the patient. You get this sudden, massive flood of new viruses released into the surrounding human tissue, which causes widespread destruction. I mean, think about it. When millions of cells in a vital organ are simultaneously bursting and dying, that massive tissue damage is exactly why infected patients experience such severe, debilitating symptoms. The cycle of infection is fully reset, ready to start all over again, spreading exponentially.
So the crucial takeaway here is this. Understanding this biological blueprint isn't just about knowing how the enemy attacks, it's about figuring out exactly how to stop it. Based on the life cycle we've just mapped out, entry, replication, or release, which phase do you think presents the best possible target for designing future antiviral medicines? If you could jam the lock during attachment, maybe halt the factory machinery during replication, or prevent that catastrophic bursting during release, where would you choose to strike? I'm going to leave you with that thought to chew on for your future studies in medicine. Thanks for joining me for this explainer, and hey, never stop analyzing the world at the microscopic level.